Okay, so good morning. Well, especially Young, who really kindly gave us the 7 a.m. slot in Korea. So thank you so much. And good afternoon, everyone. So today we are, this is the agenda of the meeting. So we are going to have a journal club. And this is a little special for us because it's a project that we collaborated with Hyeyoung Shin. That is now just published in Nature Neuroscience last month. So Hyeyoung Shin used to be PhD students with Chris Moore and Stephanie Jones at Brown, and then went on to make a postdoc with Hillel at the SNCC lab at Berkeley, during which we collaborated on this project. The reason we're having this journal club is first because we think it's an amazing piece of work, and so we wanted to help spread it out. Is also because there is interesting connections with an ongoing project that we are doing this year on predictive processing. And through this process, we made a review with more than 50 co-authors. These review help us highlight mechanisms that are involved in predicting the next stimulus. And so in the review, you can see that these are all of the OpenScope projects that we did in the last half a decade. And the Illusion project today is interested in recurrence. And I also think there are interesting overlap with potentially roles of inhibitory neurons that maybe Young will speak about today. And so what we saw through the review is that all of these mechanisms were actually somehow tackled by individual OpenScope project independently. And what we did through the review is try to highlight the shared threads that are potentially overlapping each other. And so today, Young is going to talk a lot about recurrence, which I think probably play a role in predicting the next ongoing stimulus. So it's such a wonderful pleasure to have you today. And so I'm looking forward to your talk, Ian.
Great. Let me share my slide.
Oh, and I want to mention, so we are recording this and we plan to share this video on YouTube. So please be aware of that. If anything occurred during the meeting that we need to edit out, well, please reach out before the end.
Okay, great. Hello, everyone. Thank you for the kind introduction. I am very excited to share my research with you today on how recurrent pattern completion drives the neocortical representation of sensory inference. As Jerome mentioned, this work was published last month in Nature Neuroscience, and it was done in collaboration with the OpenScope team at the Allen Institute, led by Dr. Jerome Lecoq. And the bulk of the work was done while I was a postdoc in Hillel at the SNCC lab with several of my brilliant lab mates, Dr. Mora Ugando, Dr. Lamia Abdeladim, and Dr. Uday Jagadisan. And since then, I have started my lab at Seoul National University, where we continue to study the neural mechanisms of perceptual inference. So the motivation for this study, as well as the overarching goal of my research program, is to elucidate the neural mechanism of perceptual inference. When neuroscientists investigate the neural code of perception, we tend to assess it the way we would assess a camera. In other words, neuroscientists tend to focus on fidelity, accuracy, and information when assessing the neural code of perception. However, the goal of perception is not accurate and faithful representation of sensory information, but rather to infer the underlying perceptual objects based on what is often incomplete or ambiguous sensory inputs. For example, if you're a deer looking at the scenery, your ability to discriminate between the orientation of this blade of grass from that blade of grass is completely inconsequential. What is paramount for survival is your ability to infer that this bunch right here might belong to a lion. So we wanted to figure out how the brain computes perceptual inference, and we knew from prior literature that perceptual inference requires the integration of sensory information with prior expectations about the sensory world. We also knew that sensory information is primarily communicated via the bottom-up feed-forward pathway along the visual hierarchy, and prior expectations are communicated in the opposite direction through the top-down feedback pathway. However, we did not know exactly how top-down and bottom-up information gets integrated at the neuronal circuit level. So to address this question, we leverage illusions because illusions have this unique property where the perceptual output is dissociable from the sensory input. In other words, we can examine a neural code and determine whether that code is faithful to sensory information or whether it encodes perceptual inference. Now, leveraging this feature, we can figure out exactly where in the visual hierarchy perceptual inference first emerges and focus in that region to dissect the microcircuit. And among illusions, we chose illusory contours because prior studies have demonstrated that many species, including mice, are able to perceive illusory contours the same way humans do. So we could dive straight into the circuit cracking mode in our study. Prior literature has also shown that illusory contour representation arises first in higher visual areas and gets fed back to primary visual cortex. While this explains why illusory contour representations are present in V1, it does not explain how bottom-up information and top-down information gets integrated. Now, you'll notice that the known connections in the circuitry form an open loop. So without an explicit description of where the recurrent connectivity arises, it's unclear how this integration of top-down and bottom-up information can happen. And the known circuitry also doesn't explain why the inference signal gets fed back to primary visual cortex in the first place. In other words, if all of the inference computation happens in higher visual areas and onwards, why should V1 receive an update about that signal? We found that V1 was in the loop because it played an active role in shaping perceptual inference. We found that a sparse group of neurons that we call IC encoders was critical for closing this loop. And these IC encoders receive top-down information about sensory inference and in turn drive neural pattern completion within V1. In other words, the specialized subset of neurons serves to locally broadcast top-down information and thereby selectively reinforce the neural activity patterns that match the brain's prior model of the sensory world. So that was our paper in a nutshell, and now we can dive into the nitty-gritty of how we came to that conclusion. First, we set out to localize where the inference was computed. So we leveraged two high-throughput neurophysiology techniques to simultaneously record neural activity across 6 visual areas, primary visual cortex and surrounding higher visual areas. The multi-neural pixels data, as you know, was collected at the Allen Institute via the OpenScope team, and I collected all of the two-photon imaging and holography optogenetics data in the Adesnik lab. First, we set out to replicate prior literature by showing that neurons with receptive fields exclusively within the illusory gap region can respond to illusory bars as if they were real bars. So we identified neurons that exclusively responded to the center illusory gap region and not to any of the adjacent positions. And we can show that among these neurons, a decent subset of neurons responded to illusory bars. And when they respond to them, they respond with the same orientation selectivity to illusory bars as they do to real bars. So this confirmed that in mouse visual cortex, we can see illusory contour representations, which was a replication of prior studies done in monkeys and more recently in mice. However, filtering out neurons based on receptive fields, filtering out these exclusively center responsive neurons left us very few neurons to work with. And you can see that we were left with 18 neurons out of about 1,000 V1 neurons collected in the NeuroPixels dataset. So we wanted a new definition of illusory contour encoding neurons that do not rely on receptive field positions and yet allows us to identify neurons that emergently respond to illusory contours. So we designed a new set of images that we call the LC images as a mix and match of the illusory contour containing images. And specifically, we took the top half of the IC1 image and the bottom half of the IC2 image to get the LC2 image, and vice versa for LC1 image. And because the sum of the parts are equal between the pair of IC images and the LC images, we can expect that a neural population that encodes the image faithfully would have equal sum of responses for the pair of IC images as they do to LC images. We can also expect that neurons that are faithfully encoding the inducing segments of the illusory bar would respond equally well to one of the IC images as they do to one of the LC images. So for instance, a neuron that responds to the bottom right segment of the image would respond equally well to IC1 image and LC1 image, and not so much to IC2 and LC2 image because the inducing segments are turned out in these images. And the same can be said for the bottom left segment, the top left segment, and the top right segment. So these were the neurons that we expected to see. And interestingly, we can also identify neurons that are responding to the illusory bar containing images in an emergent fashion. So these neurons respond to one of the IC images, but to neither of the LC images. And when we look at the orientation selectivity of these neurons, we can see that the orientation selectivity of the illusory bar again matches the orientation selectivity to real bars. So this identifies illusory contour encoding neurons, and we can quantify the proportion of IC encoders across all of the recorded brain areas. And when we do this, the results were resoundingly inconclusive as to whether a brain area encoded illusory bars or not. While we see a higher proportion of IC encoders in layer 2-3 of B1 than layer 4, we concluded that this was not really a good way of determining whether a brain area encoded the illusory contour inference. So in order to look at the population activity pattern in a more comprehensive manner, we turn to neural decoding. And we can decode the visual label from the neural activity pattern. And we see that when we do that, we get a high cross-validation accuracy, discriminating the visual activity patterns evoked by these 4 images. While the cross-validation performance confirms that, that there are distinct neural activity patterns evoked by the 4 images, it cannot tell us whether the inferred illusory contour is represented in the neural activity pattern. So to address this, we designed yet another set of stimuli that we call the TRE images, because it contains a T shape and it also contains real edges in the place of the illusory contours in the IC image. And we designed these TRE images such that it would have equivalent overlap with IC1 as it does with LC1. And to demonstrate this, I'm going to show the images in sequence. This is our IC1 image, and this is our TRE1 image. And you can see that the overlap is between these bottom left 3 segments of the images. And between the TRE1 image and the LC1 image, there is an overlap between the bottom right 3 segments of the image. So if we calculate the pixel overlap between TRE1 and IC1, it is exactly the same as the pixel overlap between TRE1 and LC1. So what can we do with these images? We can construct the decoder the same way as we did before to distinguish, to classify the neural activity patterns evoked by the IC and LC images. And now we can have the decoder classify the neural activity pattern evoked by the TRE images. And if the neural population is faithfully encoding visual information within the visual images, then we can expect the decoder performance to simply reflect the overlap between the images. And because there is equivalent overlap between TRE and IC as there is between TRE and LC, we can expect that the TRE evoked visual activity pattern would have an equal likelihood of being decoded as IC as it does to LC. And in contrast, if the neural population did indeed represent the illusory bar, what we can expect is that the representation of this illusory bar would be similar to the representation of the real bar in the TRE image. And that would endow an extra similarity between the neural activity patterns evoked by IC1 and TRE1 above and beyond the similarity between the activity patterns of LC evoked activity pattern and TRE evoked activity pattern. So we can expect a decoding result like this, where TRE is more likely to be decoded as IC than as LC. And that is what we see in V1 of the mouse. And when we look across areas, we see that among the higher visual areas, LM is particularly good at representing the illusory contour Now, comparing layer 4 and layer 2-3 of V1 using transgenic lines and two-photon imaging, we can see that the inference signal is present in layer 2-3 of V1, but not in layer 4. So this shows that layer 2-3 of V1 is the first station in the visual hierarchy that encodes the illusory contour inference. So now we can zoom in V1 layer 2-3 and figure out how bottom-up sensory information gets integrated with top-down inference signals. But to do that, we needed to first identify the key players in this microcircuit. And to this end, we needed to integrate the neuron by neuron univariate analysis with the population level neural decoding analysis. And so what we did was to zero out subsets of neurons, functionally define subsets of neurons in the input to the decoder, and conversely to decode from functional subsets instead of the whole population. And we can compare the decoding performance for the different subsets with the default decoder, and we can compare the cross-validation accuracy as well as the inference decoding performance. And what we see is that when we nullify the IC encoders in the input to the decoder, the cross-validation accuracy is not impacted by much, but we no longer get a significant inference decoding performance. What this means is that IC encoders are essential for mediating the illusory contour representation. And we see that when we decode only from these neurons, even though it's a small subset of the neurons, we actually get an enhancement in the inference decoding performance, meaning an enhancement in the signal to noise ratio. And when we nullify the segment responders in the input, we don't get an impact on the inference decoding performance. And when we decode only from these neurons, even though it's a larger subset of neurons than IC encoders, we lose the inference decoding performance when we decode only from these segment responding neurons. So segment responders are largely responding faithfully to the images, and IC encoders are responding emergently to the images and mediating the illusory representation found in this inference decoding analysis. So now we know that the illusory contour encoding circuit is comprised of IC encoders and segment responders, and that these are the key players. So now we're going to use two-photon holographic optogenetics to probe the effective connectivity between IC encoders and segment responders. Now, two-photon holographic optogenetics is this cool twist on optogenetics that uses holography to shape light into cell-sized balls that you can direct to any cell of your choice. Essentially, it allows us to quote-unquote play the piano with the brain. In other words, you can inject arbitrary spatiotemporal patterns of neural activity in vivo at will. And so we designed the two-photon holographic optogenetics experiment as follows. So within one session, we have an imaging block and a holography block. And in the imaging block, we're showing images containing illusory bars, as well as a bunch of other images, including the LC and TRE images that I mentioned previously. And in between the two blocks within the experimental session, we are doing an online analysis to identify the IC encoders and the segment responders. And we can do this in about 10 to 20 minutes. And having identified the IC encoders and segment responders, we can stimulate them using two-photon holographic optogenetics in the absence of any visual inputs. And we can see what that stimulation does to the rest of the network, how the rest of the network responds to selective stimulation of these functional subsets of neurons. And this shows that the stimulation works as intended. We can selectively stimulate functional subgroups of neurons, and we can identify about 20 IC encoders in each orientation. And we can identify slightly larger group of segment responding neurons that respond to the inducing segments of each illusory bar. So with this experiment, we can now do an analysis where we construct the visual decoder as previously, where we try to classify the visual-driven activity pattern according to the visual label. And we use this decoder now to classify the holographically driven neural activity pattern. And we can assess which visual-driven activity pattern is most resembled by the holographically driven neural activity pattern. And what we can expect when we decode from all neurons is that when we stimulate IC1 encoders, we can expect that neural activity pattern to be decoded as IC1, because the activity of IC1 encoders is unique to the IC1 image, and IC1 encoders are not active when shown with IC2 image or LC1 image or LC2 image. So we expect the stimulation of IC1 encoders to be decoded as IC1. And the same is true for IC2 encoders. And we can confirm that this is the case in our holographic experiments. And now we were interested in looking at the network effect of the holographic stimulation. So what we did was decode from the rest of the neurons, excluding the neurons that were directly photoactivated by the holographic light. So when we decode from non-photoactivated neurons, what we can expect is that if these IC encoders drive a network effect such that a pattern completes the visual evoked activity pattern, we can expect the IC1 encoder stimulation to be decoded as the IC1 image when decoding from non-photoactivated neurons. And that is indeed what we see when we stimulate the IC1 encoders. We can see that IC encoders drive neural pattern completion within V1 layer 2-3 in the absence of any visual inputs. This was not the case for segment responders. So while decoding from all neurons does successfully decode into the corresponding image labels, decoding from non-photoactivated neurons results in a default decoding performance. None of the decoding performances are significantly different from chance. And this is because segment responders stimulating about 20 to 30 segment responders is not sufficient to recruit a network effect that leads to a decodable activity pattern. So in conclusion, we find that the effective connectivity from IC encoders to the rest of the IC responding neurons is stronger than segment responders to other IC responsive neurons. And What this implies is that IC encoders drive pattern completion of IC representations in V1 layer 2-3. So now we have an updated model where the IC encoders receive top-down inference signals from higher visual areas and propagate that information within V1 layer 2-3. Now, while this was cool, one thing that left us scratching our heads was the result that driving segment responders did not recruit IC encoders. We initially expected that driving segment responders would also lead to a pattern completion effect, because if you look at this loop circuitry here, the segment responders communicate first to higher visual areas and propagates back to V1 IC encoders. So we thought that driving segment responders would recruit IC encoders. Now, this was not the case, and our interpretation of the negative result was that stimulating only about 20 to 30 segment responders was not enough to get a full reverberation up and down the visual hierarchy. And if this interpretation is true, we should at least be able to see decodable IC representations in higher visual areas when photoactivating V1 segment responders. So to test this prediction, we leveraged two-photon holographic mesoscope that was invented by Dr. Abdeladim with the help of Dr. Jagadisan and myself. And we constructed the holographic pathway in the Thorlabs mesoscope so that we could stimulate one brain area while monitoring the downstream effects in multiple downstream areas. So this is that technique in action. We can see the stimulation sequentially in V1, then PM, then RL. And we can see and monitor the neural activity pattern in all of these visual areas during the stimulation and shortly after the stimulation. So now, for the first time, we can probe interareal communications in a causal manner. And we leverage this technique to stimulate the V1 segment responders while monitoring the activity not only within V1, but also in higher visual areas. And indeed, we see that when stimulating segment responders, we don't see a decodable activity pattern in V1, but we do see an above chance decoding performance in higher visual areas. So higher visual areas do not include the stimulated neuron. So what this implies is that segment responders indeed send feed-forward information that allows illusory contours to be represented in higher visual areas. This result also implies a division of labor between IC encoders and segment responders. IC encoders drive pattern completion within V1 layer 2-3, whereas segment responders drive feed-forward activity. So there's a division of labor between faithful neurons that drive feed-forward activity and top-down driven neurons that mediate recurrent pattern completion. So in conclusion, we found that selective photoactivation of V1 IC encoders is sufficient to drive pattern completion of visually evoked illusory contour representation in V1 layer 2-3. And we think that this recurrent pattern completion may be an integral step in perceptual inference more generally. So in my lab, we are doing some exciting follow-up of this work, and we are reinterpreting the neural code of perception based on the perceptual inference framework at the algorithmic and the implementation levels. So if you liked my talk, please come check out our posters at SFN this year. And with that, I'd like to thank my lab members as well as my funding sources, and most importantly, to the collaborators of this work. And thank you for listening.
Thank you so much. I have amazing threads of little pieces that come together in the end. I have questions on temporal succession of events. Because if you in the NeuroPixel data, have you tried to look at the timing of responses in all of those areas to see if those really succession of events are present in the spikes?
Yes, that's right. I did look at that. Let me see if I can pull up the supplementary data. It's in our paper, buried somewhere. So we basically confirm, sorry, wrong PowerPoint. We basically confirm what Lee and Nguyen found in monkeys, which is that the representations arise first in higher visual areas and then comes back to V1. And we can even see that segment responders respond first and then higher visual areas and then IC encoders. So we see that sequence, but the sequence is very quick. So it's like only a couple milliseconds apart. And the rest of rest is the recurrent activity in the hierarchy. So presumably there's a lot of time for activity to go back and forth and iteratively refine the representation of the perceptual inference.
I see. And what do you think is going on with inhibitory neurons in that business? I'm curious about having your opinion.
Yes. Yeah, sorry, I didn't get into that. I did record from interneurons and also identified interneurons in the NeuroPixels data. There's some complexity there because I didn't mention, but we recorded illusory contours both as light edges and dark edges. So we had the black and white flipped version of the images. And basically, the selectivity in the interneurons are largely largely, responding differentially to the black and white flip instead of like responding selectively to illusory contours versus real bars. We do see that like somatostatin neurons tend to be more faithful to the real bars. And we think that a VIP, because of their interesting disinhibitory role, might have an interesting role in this process, but there is definitely more to be studied there.
Okay, that makes sense.
Yeah.
Any questions in the audience?
Anything in your current lab to follow up on this?
Yes. So we are doing less work directly on illusory contours because what I'm most passionate about right now is to find out whether this recurrent pattern completion is actually a computational motif of perceptual inference. So we're looking in different inference contexts, for example, object recognition and tracking and stuff like that.
What are you training the mice to recognize?
Sorry?
What kind of object recognition are you looking at?
So we're starting with 3D objects that has been shown before that mice can discriminate to some extent. So that's where we're starting, and we're also trying to do behavior to actually demonstrate. We were trying to actually do an object change detection task, kind of motivated from the image change detection task that the Allen Institute has done. That's really, really hard to train, as you can imagine. So we are in the process of working that out.
There were a lot of interest online on schizophrenia. I was actually surprised. Is it something you're planning to follow up, or are you still going to focus on the object part?
Yes. So my primary interest is like in the computational aspect of perceptual inference, how that happens in the healthy brain. And then I think there is obviously implications for mental disorders, but I think like using mouse models to study that is kind of far down the line. It's definitely a future direction that I might be interested in, but currently not a primary focus, unfortunately.
I see. And unless there are more questions in the audience, those data set are public. I suddenly got a few people asking me about the 2-photon data. Are you planning to release it, or?
Yes, so the process data is in the code ocean. I was having trouble trying to figure out how to upload the really large data set because like there's not an easy platform to do that. So interested people can contact me directly, and I'll be happy to send that.
Vivek?
Hello. Sorry, can you hear me?
Yes.
Yes. Oh, awesome. Okay, so really great talk. Thank you so much. It's really exciting to stimulate targeted cells and then to be able to measure the response in the other cells. And you showed that you could decode, to some extent, the kind of recurrent pattern completion meant that you could decode from the non-stimulated cells. But do you have any more detail about the dynamics of the non-stimulated cells? Like, are there very strong responders, or it's sort of a weak effect that's distributed across many cells, or?
Yes, that's true. That's also in the extended data figure. I'm going to share that with you. Hopefully I succeed this time. Okay. So what we can see here is that when we drive IC encoders, we do get, a bump in activity with the IC responsive neurons. And then the non-responsive neurons don't respond as much. And so this results in a decodable activity pattern. Obviously, the scale of photoactivated, the network effect of photoactivation is much smaller than the visual responses. You can see that the visual responses are about is an order of magnitude higher than the photostimulation driven effects. And this, again, is because we're only driving about 20 neurons. And this is, we estimate to be a very small fraction, like 1% of the entire local recurrent network in V1 layer 2-3.
Sorry, so you're pointing, I'm kind of getting lost in the figure. Are you pointing to D and E? No.
Sorry. So we do get, mean responses. We do get the driven activity in the traces.
Yeah. Sorry, where should I look? Sorry for like the non-targeted cells response.
Right. Oh, sorry. I didn't realize that you can't see my mouse. I'm using the laser pointer
now I see
now.
it. Yeah.
So yeah, sorry. Yeah, so when you drive IC encoders and IC2 encoders, you see a higher activity pattern, higher activity driven for the image responsive neurons than the non-responsive neurons, non-visually responsive neurons. Yeah. But
is
yeah,
that
this
0.05, is that a
small.
big fluorescence z-score change? That seems maybe
No,
small.
it's tiny.
Tiny,
It's
yeah.
an order of magnitude smaller
Yeah,
than the
that's what you were
visual
saying.
responses, which is what's shown here.
Right.
Yes.
So do you imagine, I maybe Josh was kind of hinting at this, or maybe this was discussed already, sorry, but like, did you try a task where mice would like have to lick if it's the illusion versus real bar or something like that?
Yes, so that's an excellent question. We didn't try that. It would obviously be great if we could drive IC encoders and get the actual like hallucinogenic effect. And it would also be great to get a suppression of that by suppressing the IC encoders. But we were we didn't try that, and I think that the actual like generative effect, the sufficiency effect of driving IC encoders and getting perception would be a tall order because we are getting such a small network-driven effect with stimulating only 20 neurons, which was, we get 20 neurons when we image 4 planes simultaneously. And so this is a sparse group of neurons. It's particularly difficult for that reason as well.
So moving forward with your own lab and all that, are you hoping that are you thinking of the holographic stimulation as more a way to probe, like you said, connections, but not as a way to manipulate the dynamics that drive behavior?
Yes, I think so. I mean, definitely we are interested in like causally manipulating behavior with holography as well. But I think that first we see the network effects and then try to design the experiments based on that to really like get an equal scale of differentiable activity patterns in the visual case as we do in the holography case.
Cool. Thank you.
Thank you. That was a great question.
Any more questions in the audience?
Maybe one question. So for the completed pattern and those neurons that get activated from the feedback, is there any kind of retinotopic relationship to where those lines would actually be, if they're there in the image?
No, so there wasn't, surprisingly. We looked at the receptive fields of these illusory contour encoding neurons. So what I mentioned previously was that if we define illusory contour encoding neurons based on the receptive fields, we get very few neurons. And when we define them based on the IC and LC images, we suddenly get a lot more neurons. And that's because the IC encoders can have receptive fields not only in the gap region, but also in other regions. So this is the receptive field positions, the receptive field mapping. And the IC encoders, the density of neurons is kind of like everywhere. It's not concentrated to the gap region, whereas segment responders have receptive fields sort of where you would expect it to be. So this is kind of surprising, but it can be explained by the fact that feedback connections are much more diffuse than feed-forward connections. So it's not really confined to the classical receptive field. So that's how we're interpreting this result.
I mean, it could also be that the classical model breaks in these cases, right? Where you're not defined now by your receptive field anymore. Is that fair to say? Or?
Yes, I completely agree. And I think that classical receptive fields, it is known that they're primarily defined by feed-forward connections, and IC encoders are primarily driven by top-down inputs. So it makes sense that those two may not necessarily match.
There's some interesting discussion I had recently with Nuno on the EM dataset, and they found that layer 2 cells are very different than layer 3 cells, and I'm curious whether you've looked into that with your data. Like, did you
said
see
between
actually
layer 2 and layer 3?
That's right, but there's a striking different connectivity, and so I wonder if you'll be able to, if you have a higher density of IC cells in layer 2. In the very superficial section of the data.
So I didn't do that analysis. I'm quite surprised to hear that. Because there's not a clear border, at least in terms of histology, right, between them. So is it more of like a gradual change between the superficial?
You should reach out, but in terms of connectivity in the EM data, it's really striking.
Oh, wow. Okay. I should look into that. I am actually also, we have a project. One of the posters is about analyzing the microns data set that you guys were obviously a part of. And yeah, we haven't looked at in that aspect. That would be very interesting to look at.
Because your pattern is very much top down, so you should probably I would expect to see those cells really stronger in the superficial section of the data. Yeah.
Yeah, possibly. Yeah.
Okay.
Can I ask another question? Sorry. Oh, sorry, didn't see that.
Yeah, please, Vivek.
Yes, please.
Okay, sorry. Yeah, I mean, the mesoscope holographic seems like crazy. Could you say more about that, both in terms of like, did it reveal a lot? I mean, just looking at the data, I don't know, when you stimulate one area, you mentioned you could decode, ascending signals to higher up. But yeah, I mean, it seems like what a crazy opportunity, right? To stim one area with ensembles and see the effect propagate over many areas. Was it, I don't know, could you describe the data, also maybe your experience collecting that data?
Yeah, no, absolutely. This was a very exciting project to be a part of, and I'm very excited about the potential of this technology. I am actually trying to build that in my lab currently.
Nice.
But with this paper, we were only kind of like using it as a proof of principle, and looking at, kind of like confirming the most rudimentary aspect of the coding circuit, which was more or less known before, but we just wanted to know why we weren't seeing the entire reverberation in the recurrent circuitry. And so that's how we used it. I wish I had more time to use it for cooler things, for actually like discovering new circuit connectivity and stuff. But yeah, that's when my postdoc ended. So I ran out of time essentially, but we are going to do that in my lab. So look out for it. Yeah.
Is that data also going to be released? The like holographic in one area, but imaging with the mesoscope?
So that again, the post-process data is released, but and that contains the trial by trial responses, I believe. But
Nice.
yeah, but because it's such a targeted stimulation, there's only so much you can glean out of that experiment, because it really depends on how you design the stimulus and stuff. So I don't think there's a lot of exploration to be done in that particular data set. Yeah.
I see. Okay. Thanks.
Okay. Any more questions for Hyeyoung? Well, thank you so much for presenting this so early from Korea. We really appreciate it, and it's a fantastic talk.
Yeah, this was so much fun. Thank you. And it's great to see a lot of my co-authors on this channel.
All right.
Great talk.
Thank you, everyone.
Thank you.